Page 41. So today we're going to take a little more in-depth look at DNA itself. Last day we looked at sort of bigger level, like, you know, it becomes chromatin, it becomes chromosomes. Today we're going to go in smaller and look at the details of the DNA. So the first thing to realize is that it is made of two strands. DNA is made of two strands, and they are wrapped around one another. Do you guys remember what this shape is called? That is called a double helix. Okay, and that's a vocab word. All right? Now, it's really, really hard to sort of do things because this is a three-dimensional structure, so it's hard to do on paper. So we have a tendency to flatten it out, okay? And sometimes we'll just take one of the strands, and that's what we're going to do right now. We're going to take one of the strands, and we're going to flatten it out so it looks like that. Does that make sense? Mm-hmm. We've taken one of the strands and we flattened it out. So there's our 2D diagram. We're going to learn a new word now. That word is polymer. What does the prefix poly mean? Many. So poly means many and mer means units. DNA is a polymer. Do you see here that we have a unit here and a unit here and a unit here? Do you see how it's got all sorts of little units that look very similar to one another and they're just stuck together in a big long chain? That is a polymer, many small units stuck together. Yeah? Now, if you look at only one of those units, what's the prefix meaning one? Mono. So a monomer is a single unit. All right, so here we have lots of monomers stuck together, and this is one monomer. So monomer is a word you need to know. Polymer is a word you need to know. They'll come in really, really handy if you take Biology 12 because carbohydrates are polymers made of monomers called sugars. Proteins are polymers made of monomers called amino acids. And there's a whole bunch of things that are just like that, okay? We have another new word, and that word is nucleotide. So we're now on our next page. Nucleotide. Say it with me. Nucleotide. Nucleotide is a single unit. So a nucleotide is a monomer of DNA. So here is our DNA strand. That is our polymer. This one little piece here, this is our monomer. That is called a nucleotide. And that is the monomer. And if we look at the nucleotide up close, you can see it's got this part that is like the side of the ladder and this part that helps make up the rung of the ladder. So a string of monomers make up a polymer. A string of monomers make up a polymer. In this specific example, the monomers are nucleotides and the polymer is the DNA strand. So if you were just underlining, you could do a 1 underline here and a 2 underline here. Now this nucleotide here, this one, is simplified. Okay, we're going to unsimplify it a little bit. It's not really just a gray block. It's actually a P, which is a phosphate. Do you guys remember phosphate? Okay, phosphate. If you don't remember, you don't need to know it for this unit in particular, but it was PO4 3-. Okay? And then we have, what kind of a, this is our circle, and we usually represent the phosphate with a circle. What shape do we have here? How many sides on that shape? 5. So what type of a gon is that? That is a pentagon. And that pentagon is the sugar. So we have a phosphate attached to a sugar. And then sticking off the side of the sugar, we have a base. So a nucleotide, the phosphate and the sugar and the base all together is a nucleotide. So do you need to know phosphate? Yes, you do. Do you need to know sugar? Do you need to know base? Do you need to know nucleotide? Yes, you need to know all of those. Now, we're going to actually do a little bit of drawing here in a moment, but not quite yet. Not quite yet. So we usually start with the phosphate at the top. We have the sugar next, and then sticking off the side of the sugar is the base. Okay? Sometimes we will also call this a nitrogen base. What is wrong with my pen? Why is it not behaving? There we go. Because this base contains some nitrogens. But if you just want to call it base, that is fine. Okay? So phosphate, sugar, and base makes a nucleotide. Now, if I get you to draw this, you're going to have to be able to draw a pentagon. Now, I'm going to show you how to draw a pentagon, and you're going to practice, because I find that in grade 9, you don't spend a lot of time doing shapes in math anymore, and so very often people cannot draw a pentagon. So to make it a little simpler, we're not going to have the pentagon tilting. Okay, we're going to draw it straight up and down to make it easier. Yes? So what we're going to do is I'm going to show you how not to do it, and then I'm going to show you how to fix it. Okay? How not to do it, you're going to draw, it's just like drawing a little tiny house. Okay, you got the sides of the house, and then you have the roof of the house. except that instead of having the sides of the house go straight up and down, you're going to make the sides of the house go closer together so that you have it at the bottom. So you're going to have two lines like that, and then you're going to draw the sides of the house going somewhat inward, and then you're going to draw along the bottom. And if I take too long, I don't actually do as good a job if I just kind of go like that. I find that if I try too hard to draw it, I actually end up with a worse result than if I don't. So practice drawing maybe 10 pentagons. Okay? Nice. Good. Please ask for a pencil rather than that's kind of useless. Very good. That's really good. Yeah. I've been drawing for years. Right. Nice pentagon sliding. It actually looks a little different though, you know? I wish it was a little skeletal. Yeah, I would like I'm making this chapter on the because I feel like it's just yes, but at least that's fair. Okay, so now what we're going to do is we are going to practice drawing one pentagon, and off the top we're going to draw a phosphate, P, and off the side, we're just going to draw a rectangle. So practice drawing one nucleotide where you have your pentagon. That's your sugar. Above it, you're going to draw your P for phosphate. And off to the side, you're going to draw a rectangle for the base. Now, if you're wondering what you should be putting on your cheat sheet, yes, you can draw pictures on your cheat sheet. Will you be expected to label diagrams on your tests and quizzes? Yes, you will be expected to label diagrams. So you can draw whatever diagrams you want to. Okay? All right. So now we have a nucleotide. Now you'll notice that we just drew a rectangle, but our nucleotides, the bases are actually a little fancier, okay? And if you remember, there are 4 different types of bases. Do you remember the 4 letters? A, T, C, and G. And one thing you're going to notice is that it's not beautiful on here, but the A is shaped like this. It's shaped like a inward triangle. The T is shaped like an outward triangle. The C is shaped like an inward circle, and the G is shaped like an outward circle. Can you guess which one matches up?
C.
C matches up with? Well, it's okay. We'll come to it later. Don't worry. Don't worry. We'll come to it later. It'll make sense. Okay?
Oh, I thought it was like the shape of it.
Yeah, the shape matches the shape. So A would match with T,
Oh.
right? Okay, guys, here is an A. Yeah, we got that inward triangle. Here is a T. We got the outward triangle. Those match. Yes? Like a baby bird. Okay? Here is a inward circle. The inward circle matches with the outward circle. So that means that A gets paired up with T and C gets paired up with G because they go together like a puzzle piece, okay, across from each other. And we'll see that very soon. Remember, if you want to know the names for the bases, these are the enriched names for the bases: adenine, thymine, cytosine, and guanine. And because there are 4 different bases, that means there are 4 different nucleotides. Yes, Spencer? What about U? U is actually only in RNA. Okay, so thymine is in DNA. U, which is uracil, is in RNA. And U takes the place of T in RNA. It works in the same way as T does. They're actually very similar in structure. There's just a very small difference. Yeah. So let's watch how these guys come together to form a polymer. Now we're still just looking at one strand. We're not going to be pairing them up. You know, we're not going to have the baby bird with the food, and we're not going to have the circle with the circle. Okay? Right now, we're just going to link up one nucleotide with another nucleotide in a line. So what we've got is we've got one nucleotide, we've got a second nucleotide, and what happens is that we just draw a line between them, and that is a covalent bond. So they stick to one another. Okay, so the covalent bond sticks the nucleotides together in a chain. So we have a monomer. We have another monomer. Now, technically, this one here, because there's only 2, we could call it a dimer, but we're just going to call it a polymer. Okay, because poly just means more than one. And this process repeats over and over and over until you get a long polymer, and that is 1/2 of the DNA strand, and it looks like what we've got over on the right-hand side. Yeah, over here. Big, long DNA molecule, which is only 1/2 of the DNA molecule, but that's okay. That's what we're focused on for now. And you'll notice that we've got all sorts of different bases along the way. What do we call the letters? Like if we were to read this, if we read this as A, T, C, G, T, T, C, G, C, A. What do we call this sequence of letters? The what? The code. It is a kind of code. Do you remember what type of code it is? Yes? You're so close. We're going to add a little something onto the end of the word gene. Genetic. Okay, so that is our genetic code. Okay, and genetic code is a vocab word, so you should know that. The sequence of letters is the genetic code. Now, we talked about this before. But we only briefly mentioned it. When you have that sugar phosphate, sugar phosphate, sugar phosphate all attached together, it provides support for the DNA. What provides support for you? Your spine, which is also known as your backbone. Yes? So just like your spine is your backbone, the side of the DNA molecule is known as the backbone. So this part here, the sugar phosphate, sugar phosphate, sugar phosphate, is called literally the sugar phosphate backbone. Okay? So the sides of the ladder are the sugar phosphate backbone. So it's this twisty part, not the rungs of the ladder, but the sides of the ladder. And sugar phosphate backbone is a vocab word, okay? And the bases all stick out the side off of the sugar phosphate backbone. There's our sugar phosphate backbone, and this is we got our bases. Base, 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 base. It is the bases that join the two strands of DNA together. So the bases join the two strands of DNA together. So what's going to happen is that you're going to have, remember we've got an inward triangle and we got an outward triangle. Do you see how those are going to match up together? If we bring them together, they're going to go, yes? Outward triangle matches with inward triangle. Inward circle matches with outward circle. Outward circle matches with inward circle. So they all match up like puzzle pieces. Yes.
Is it possible for them to somehow look at mix up the colors?
No. What will happen is if you, like, let's say you had a mutation. Logan, let's say you had a mutation, which would mean that maybe instead of having the proper matching one here, maybe we ended up with a outward facing one, right? Outward circle. That would cause a mismatch and they wouldn't bond properly to each other. And so what the DNA would do is there are repair mechanisms. So it would either come in and repair it correctly, right, back to the inward triangle one, or it might repair this one to the new one, and then you have a mutation that has stuck. Yeah? So you can have mutations that get fixed, and you can have mutations that stick. Yeah? Ken? What is the base made out of? That's a very good question. I think I have a picture of it somewhere. Just give me a second. Actually, you know what? Hang on one sec. Yes. Okay. DNA structure. That is thymine. And at each corner of that hexagon, you have little carbon atoms. Carbon, carbon, carbon. Nitrogen, carbon, nitrogen. So crazy, yes? You don't need to know those structures. But what you can see is that you get these like little intermolecular forces in between and they match up perfectly. And rather than having like a triangle shape, these ones form, the A and the T form 2 bonds, the G and the C form 3 bonds, okay, or intermolecular forces to be more specific. You don't need to know that level of detail, but if you're interested, that's what's going on. And you'll notice that the adenine is kind of bigger, like it's longer, and the guanine is kind of bigger and longer, and the thymine and the cytosine are kind of smaller. That way the length of the rungs are always going to, if this one is the longer one, this is the shorter one, this is always going to be the same length as one that's got a longer one and a shorter one. You always have to have long with short for them to all be the same length. Okay, again, you don't need to know that. You can just be like, okay, yeah, Miss Spence, whatever. All right? Okay. So these guys match up, and this is an enriched thing, but what do you notice about the sugar phosphate backbone over here compared with the sugar phosphate backbone over here? They're the other way. One is upside down. Yes? So this one goes sugar, phosphate, that one goes phosphate, sugar. Yeah? But they do line up like this, so they are parallel, yes? Can we agree that those two things are parallel? Yes? If we take one though and we turn it upside down, we actually call it anti-parallel. So that's an enriched word, if you like. Because the strands are upside down relative to one another, we say that they are anti-parallel. That is an enriched word. Okay? The bases pair up, and they are held together by something called hydrogen bonds. Hydrogen bonds. Hydrogen bonds are a type of They're bonding between molecules. What do we call those things? IMFs. So they're a type of IMFs. And I don't know if you remember, but IMFs are intermolecular forces. Are they strong or are they weak? They're weak. Okay? But out of all the different intermolecular forces out there, hydrogen bonds are actually the strongest IMF. This here is enriched. Hydrogen bonds are the perfect type of force to hold the strands together. Because if they were really strong, like a covalent bond, you could never get the strands to separate. And you need the strands to separate so you can make a copy. Okay, we'll talk more about that later. Okay? You need them to be weak enough to separate, but you also need them to be strong enough to hold it together stably. And since hydrogen bonds are the strongest IMF, that means they are just weak enough and they are just strong enough to perfectly do their job. They're perfectly good staying together, but if you give them a little kick, they can come apart. There's little enzymes that'll push them apart when they need to be pushed apart. Okay? But that little bit there, describing why they're perfect, that is enriched. Okay? That breaks them. We're not going to go into too much detail on that. We'll look at it a little bit. There is an enzyme that comes in and breaks them. If you take Bio 12, they will go into big detail on that. They'll be like, there's the RNA polymer, or the, what do they call it? I can't remember what it's called. It's been so many years. But there's an enzyme that goes in and splits them apart. And then there's another enzyme that comes in and like brings in the nucleotides to make the copy. Yeah, it's very cool. Where have you seen this? Yeah, it's very cool. Yeah. Now, we're going to learn a new word. Oh my gosh. How many new words have we learned? Like a lot. Okay, so here's our new word. The word is complementary. This is an E here. Not complimentary. Complimentary would be like, wow, I love your hairstyle. Or those are great new boots. Okay, that would be complimentary. Complementary means things match up perfectly. Complementary means to match up perfectly. This is a vocab word. And so you remember we had our triangles and then we had our circles. The triangle matches with the triangle. The circle matches with the circle. The ingoing circle matches with the outgoing circle. The outgoing triangle matches with the ingoing triangle. So A always matches with T and C always matches with G. And this is what we call the rule of complementary base pairing. This entire thing here is a vocab. A goes with T. G goes with C. Now, how are you going to remember that? When I was in high school, all I remembered is that C and G kind of looked like the same letter. Okay. But you can remember that, where do you find apples? Apples are up in the tree. So A goes with T. And where does the car go? The car goes in the garage. So C goes with G. We can use the rule of complementary base pairing to figure out what the complementary strand is. So if I give you one strand, if I give you the right-hand strand, you can figure out the code on the left-hand strand. So if I give you the letters of one strand, you can figure out the letters on the other strand. Shall we try it? So here's DNA. This is our DNA here. That's one strand of the DNA. And we have A, G, T, G, C, T, T, A, C. Can you figure out what the bases would be on the complementary strand? You want to try drawing them in? Can you try? Remember, apples go in the tree and the car goes in the garage. Try it. See if you can get the letters on that complementary strand. This is on the test too. Yeah. Okay. We're going outside. This is exciting. Pardon me? Yeah, you can grab your phone if you want to.
First time. Is this real? Is this real?
It's not.
How would you make that happen?
Yeah.
Listen, we go out, we go into the parking lot and then down the driveway, okay? Turn left at the stairs, left at the stairs, okay? Look at the stairs, okay? I'll have it way out on right. Come see me. Someone.
Oh, I don't think you know, I don't, the ball guys, how we do that.
No, they highlighted.
Hi.
No, for nothing.
Can I just go to the washroom and grab a passport, Jennifer?
Yeah, yeah.
I also thought that finance had a. Yes, and when I also heard it was supposed to be part of. Not someone spoke for fun, right?
Hi, Lisa.
Oh, yeah. Oh, okay. That's fine. Oh, I think I already did those pictures. Wow. That was fine. Oh, I did most of it. I just did like one question.
Yeah.
Oh, wow. We did it for every test.
Okay, folks, we got sort of cut short. So I do want to give you your homework before we completely run out of time. It's going to be actually going back a couple of days to what is DNA and how does it work. You're going to do pages 25 to 27. Okay, and then the cheat sheet, you're going to go through, and do you remember how we starred a bunch of words? In there, you're going to get your cheat sheet from pages 16 to 27, which is all of 5.2a. So you'll be like, protein, ribosome, amino acid, those ones. Yes? So you're going to start adding those in there. All right? And I might even check your cheat sheet next day because sometimes people are like, oh, she doesn't check the cheat sheet. It's okay. I don't have to do it. I'll just photocopy my friends when it comes to the day of the test. Well, let me tell you, I will be looking to see if yours is photocopied.
It's just like super.
What do you mean if it's too good? You can do however well you want to. You can just you can write all the answers to everything. It might be better to pare it down and organize it, right? Because otherwise you're looking for things and you don't know where they are. But yeah, I don't mind. Really, anytime you write something down, it goes into your brain. Yeah? So basically, I'm tricking you guys into studying, is what I'm doing, right? By saying, here, make a cheat sheet. Well, and you know what? Honestly, if you want to write the test without your cheat sheet, you can get enriched for writing your test without your cheat sheet. So if you get to next Friday and you're like, I feel confident because I have made this cheat sheet so well, you can do that. Okay? Up to you though. You can start without it, and then if you're like, oh, I need it, you can go back and get it later. Okay? Oh.
So I didn't start my cheat sheet yet, but then today I have to record a
It's
picture.
okay. I know you're a good student. You'll get it done. Okay? Yeah.
Hey, what are we doing with our phones? We're not on our phones. We're still in class time, no phones. I know you've got them with you, but put them away.
Yes. It's up there, yeah.
Oh,
What?